Thank you for joining us. I'm Karen Utley. I'm with the International Foundation for CDKL5 Research, and we appreciate you joining us on a weekend. I will say this is being recorded and it will live as a resource for those who maybe have to drop off to take care of their little one or some other reason. You will be able to see this later. We are gathering today to hear a little bit about some research opportunities for Family Conference, but this is not just for the people who will attend conference, because it's always great to learn about research and efforts that are going on in the CDKL5 space. We are joined today by Massimiliano Bianchi from Ulysses, Wendy Gold from University of Sydney. Oh, Alice, I'm going to mess your name up. Pinato, I asked, but I will let you reintroduce yourself if you need to with the correct pronunciation. My apologies. And Connor Maltby from Ulysses Neuroscience also, and Peter Augustine, who may or may not be on camera. Thank you all for joining us. I'm going to be very respectful of time, and I'm going to pass it off to Mossy to give his presentation, and then he will pass it off to Wendy, and then we'll go from there. There is a Q&A box. If you have questions, please feel free to put your questions in there, and we will have some time at the end to answer those.
Okay, thank you, Karen. First of all, thank you to all the attendees. It's great to have you here. Thank you for taking your time on a Saturday. This is great, and it's great to have all of you attending this webinar. Actually, before starting my presentation, I would like to share with you a video, and the video is a testimonial from families that have already participated in the study, in the biomarker study that we are driving, which is sponsored by the Reboot Foundation, and it's called Elpis. The families that you will see giving their testimony are the families that have participated in what we have called the East European Elpis Family Day, which took place in Bratislava, and a lot of kudos to Peter Agustin, who has actually made all the efforts for making that event in Bratislava, in Slovakia, happening. We also had families from Czech Republic and from Bulgaria. I will tell you more about this event. I will tell you more about the science that is behind Elpis and what the project is about, and how this project will be integrated in the Family Day that you will have in Philadelphia in June. Connor, can you try to play the video?
Absolutely. So let me know when you can see my screen.
Hello everyone, my name is Peter Augustin. I'm from Slovakia, a small country in Central Europe. I'm the father of an 11-year-old boy with CDKL5 mosaicism, Peter, or Peťko. Today, I want to speak directly to the heart of this community, the patients, the parents, the siblings and the relatives who stand together in the face of rare disease. Living with a rare condition can sometimes feel isolating, but every one of you here represents strength, resilience and hope. And today, we have an opportunity to turn that hope into action. By allowing your child to participate in this clinical trial and by providing a simple blood sample, you are contributing to something much bigger than a single test. You are helping researchers better understand the disease, improve diagnosis, and move closer to safer treatments. And one day, perhaps even a cure. That is why we decided to take part in this Elpis study. Although we are a small country with only 6 diagnosed cases, 2 of them are living abroad, we connected with families from other countries, the Czech Republic and Bulgaria. These families did not hesitate. They flew and drove to our hospital in our country, resulting in samples being collected from 9 CDD patients and 17 controls. As you may know, we also need a control group consisting of healthy volunteers. Please do not hesitate to participate and encourage others as well. Siblings of children with CDKL5, cousins and friends. Progress in rare diseases only happens because of brave communities like this one. Every sample is a step forward, especially with an ultra-rare disease like CDKL5. Every participant is a partner in discovery, and every family that joins brings us closer to a future with better care and more answers. You are not just participants in the study, you are pioneers of a progress. Thank you for your courage, your generosity, and willingness to help shape a better future for everyone affected by this rare disease. Please now listen a few words from the participants from each country.
Здравейте! Аз съм Илияна, а това е
Калина.
силно Калина от град Варна, България. Включваме се да ви агитираме да вземете участие в изследването Elpis. За нас беше много важно да подкрепим това изследване, тъй като по този начин ще се научи повече за CDKL5 гена. Искаме да изкажем огромни благодарности на Петър и Саша и всички хора, които бяха
Пепи.
там, и Пепи, да, в Братислава. Всичко беше на ниво, почувствахме се безкрайно спокойни и сме благодарни на топлото посрещане. Искаме да ви кажем, че няма нищо страшно. Вземете участие, така ще се научи повече за заболяването на нашите деца. И сега едно огромно благодаря на фундация Лулу. Без вас всичко това нямаше да бъде възможно. Благодарим. Чао от нас. Кажи така. Така. Дай целувка.
Ahoj, já jsem Lucie, tohle je moje dcera Zuzanka, která je CDKL5 pozitivní. Pocházíme z České republiky, což je malá země v srdci Evropy. Jsme moc rádi, že jsme se mohli zapojit do této studie. Potěšilo nás, že se o problematiku této nemoci vůbec někdo zajímá. Přispět k dalšímu poznání této nemoci jsme mohli pouhým odběrem krve, což byla pro nás maličkost. Věříme, že tento výzkum bude jedním z kroků k pochopení této nemoci a pomůže k následnému nalezení léků, v což všichni věříme. Tak se k nám přidejte.
Здравейте, аз съм Венелина, това е Теодора. Когато разбрах за проучването Elpis, веднага поисках да се включим и да участваме цялото семейство. Смятам, че по този начин пряко участваме в намирането на нови терапии и по-успешни лечения за заболяването. Включете се и вие. Заедно можем много.
Ja som Jarka zo Slovenska, som Peťková mamina a svojou účasťou na Elpis Study pomôžete zvýšiť poznatky o tejto zriedkavej chorobe, tak smelo do toho.
So I have to say again, thank you. It was very touching for us, so I have to say thank you again to all the families that have contributed to put together this testimonial. The community is everything for the Elpis study. So without the community, we cannot really perform the study, and without the community, there will be no Elpis, and actually there is an Elpis. And I will give you an update on where we are now. We have made incredible progress, and this is really thanks to you, thanks to all the CDKL5 families that participate into our study with such motivation, dedication, and such hope. Elpis is ancient Greek. This is the name we have decided to give to our project. In Ulysses, we have this habit to give to our most important project names of gods or goddess coming from mythology. Normally, we choose the Roman Greek mythology, but we also can go into other folklore. Elpis was the goddess of hope and was representing hope itself, and this is why we have chosen that name. I will try now to share my slides. We have tried before, we have tested it, so it should work. Okay. So Elpis is an international global biomarker study which is aimed to collect
blood samples that are then separated into plasma. So the plasma is separated from the blood from the CDKL5 deficiency disorder patients. It is a study that has now run for 3 years. I will give you an update and I will show you how expanded is now the study. And again, the objective is to identify biomarkers of disease progression in CDKL5. This is a study that is only possible thanks to the collaboration and relationship we have with the patient community. Without you, this study is not possible. So we hope you will join this study and you will be a key contributor to research in CDKL5 into new discoveries because we need it. We really, really need it. If you want to be part of the study, wherever you are in the world, you can contact us at the email address that is shown in this slide. So it is already a few years that Elpis is running. It has been a learning curve, but we have now a very nice established workflow for Elpis. So how does it work? So as I said before, first of all is the connection with the patient community. We connect with you, we talk with you, and it is you indicating to us the clinician that can help to collect the samples. It is you indicating us the site for collection. And the site for collection can be an hospital, or can be more hospitals in one country, or can be a family event, as the one that we run in Philadelphia for IFCR on the 18th of June. It is actually now more popular between the patient community to go with family event because it's a nice way to gather all the family together. And we are very well equipped to have pediatric nurses coming during the event and collect the blood. And then we have labs supporting us to separate the blood into the plasma. If we don't do a family event, it is also part of the workflow to take care of the regulatory and contractive stage. This is the most difficult part of all Elpis, which is really dealing and negotiating with both the clinician and the hospital. After this is done, there is an ethical submission. So to collect blood from your kids and from controls, healthy controls, we need to have an ethical submission. So we need to write a protocol that has to be approved by the local ethical committee. Now, between the regulatory and contracting stage and the ethical submission and approval, as I said, this can be long, sometimes difficult, even if now we have a very standardized way to do it, but it can take even 6 months, if not longer. When this is done, we then send to the site that has been planned for the biocollection our clinical kit. And this is another very important aspect of Elpis. What is a clinical kit? A clinical kit is essentially a box that arrives at the site. Inside the box, there are vials for blood collections, the vials that are very much needed for our biomarker analysis. So these are vials which have a special component inside, which is the one that we need. What is also very important is that the vials from where the plasma will be then transferred are barcoded, and this barcoding system will allow us to track the samples wherever they are, wherever they go, and also to track the patient. So the patient becomes a barcode and everything goes into a very good software system which allows us to track everything. The reason why this is so important is because by sending those kits to all the different sites, all the sites will use the same consumables, the same materials, will follow the same protocol. And this is extremely important to decrease variability in a biomarker study. And this is why, one of the reasons why Elpis is so successful in the results that are then produced. There is then the patient recruitment, where the help of the patient community is fundamental. So, trying to have as much patient as possible. And also very important is to have the healthy control. The healthy control, the ideal healthy control has to be the same gender and more or less the same age. It's what is called gender age matched. And this is very important to have reliable data. After we collect the samples, there is the shipment of the samples, which is under control conditions. We use our own courier. Samples are shipped around the world. Well, they're shipped from the site to Dublin when we do the biomarker analysis. They're shipped in dry ice, but inside the box, there is a little thermometer to control the temperature during the shipment. So we know exactly the shifts in temperature that have occurred. And so this is a way for us to control the quality of the samples received. Once the samples arrive, they're biobanked at a highly controlled -80 centigrade facility. And then we do the biomarker analysis, and then we produce a study report, which means we disclose the data that we have obtained. Our labs at Ulysses Neuroscience are equipped with cutting-edge equipment and technologies for biomarker analysis. We don't only work with CDKL5, we also work with many different brain and neurological disorders, from ALS to MS, Alzheimer, Parkinson, Charcot-Marie-Tooth disease, schizophrenia, Fragile X syndrome, depression, and we have all developed and validated a series of different biomarkers. Our instruments, you can see them on the slides, are mainly three. One is called Odyssey, which is a pretty nice name for a company called Ulysses. We have another instrument that is called Mesoscale or MSD, and another which is a new acquisition only 6 months ago, which is called Luminex 200. All those technologies are complementary between them and allow us to analyze multiple biomarkers in one single run. What is also important is that all our assays are cross-species translational, which means that all the assays that we develop can be used for rodents, can be used for humans, can be used for non-human primates, and can be used for dogs as well. When it comes to Elpis, Elpis is a study that is hypothesis driven. So we have an hypothesis, and so we are targeting some specific biomarkers to analyze, and one of those biomarkers are part of a system that is in the neuronal cells, which is called cytoskeleton. And I will tell you more about this in a minute. We analyze two proteins, the acetylated alpha-tubulin and the tyrosinated, detyrosinated alpha-tubulin. We will focus more on the acetylated alpha-tubulin. We analyze also another protein, which is called BDNF, brain-derived neurotrophic factor, which is very important for promoting neuronal plasticity and development of the neuronal cell. Neurofilament light is another protein that is expressed in the brain, but it can be found like BDNF, like cytoskeletal proteins in the plasma. Neurofilament light has been found increased in neurodegenerative diseases, but also in diseases characterized by epileptic seizures. I want you to pay attention to this diagram that you can see on your right-hand side. You can see there is a neuron on inside the head, but there is also another neuron that is receiving messages and is then going throughout the body with this prolongation, with this axon, if you want to say this. It is important because the brain and the body are highly connected. If something is altering the brain, it's also altering the rest of the body. And we know that CDKL5 deficiency disorder, CDD, unfortunately, is not only a disease of the brain, but it's also a disease of the rest of the body. This is why we also check a panel of inflammatory markers that give us an idea of the conditions of the immune system in CDD patients. Okay, so where we are now, we have started with a first study, which we called Elpis 1. In Elpis 1, we had 7 sites, probably even more. The sites that you have in the green tab tables are the ones that have completed the collection. We had a few sites from Italy that in general is the country that has collected more samples in different sites. We have collected samples in Ireland 2 times. We have collected sample in Portugal, and a very small set of samples from a naturalistic study that was already ongoing. So this is not something that we have done directly, but we acquired samples already connected. Anyway, I have to say a big kudos to the day that we have done in Slovakia last year in December. because in one single site in Eastern Europe, we were able to have patients from Slovakia, Czech Republic, and Bulgaria. And we were able, in one single go, to have 9 patients and 17 healthy controls. We have to say that Brazil has also started collection, started at this time as we talk, they are collecting. And we also got ethical approval. Finally, it took a long time for Paris and Madrid, so in France and Spain. And we expect to get another 10 participants each. So what is the target? The target is to arrive to 100 samples. We have collected 49 so far, so we're halfway there. So we're very happy about this. But how to reach 100? Well, we are expanding to what is called Elpis 2 and expanding to Elpis 3, which is of great interest for you for this webinar, which is the collection of samples during the IFCR Family Day. Concerning Elpis 2, we have decided to expand beyond Europe. So Elpis 2 is very much focused on South America, with Peru, Colombia, and Argentina that will start collection very soon, actually in the Family Days. We have also added another site in Spain, in Barcelona. We are still discussing with Israel, problems. There are some problems in there, so we'll see if this site will be able to start. Germany is very well advanced. A Family Day has been already planned for September, so we're looking forward to go there and do some bio collection with our German friends. It's not finished. We have also some new additional sites, Australia, which is already collecting samples, and Tokyo. And this is new and it's a great addition. And this will happen during the Family Day in Tokyo on the 4th and 5th of July, where we will collect another 10 samples. This is a map of how Elpis looks now. We want more probably. There is one flag and one country that is missing, which is United States, and this is why Elpis 3 has been decided. Elpis 3 is the bio collection at the IFCR Family Day from the 18th to the 20th of June. Our target is to collect around 25 CDD samples and around 15 healthy controls. This study is only possible thanks to the generous sponsorship of the Lulu Foundation. Where we are now, we are finalizing the submission of the ethics. We are progressing extremely well in there. Also, all the logistic has been put in place. We have 2 pediatric nurses that will come for collecting blood from your kids. The sample collection will happen during the Family Awareness Day. The clinical kits will be sent at the place of the event. We have also engaged a lab, which is not far from where the event will take place. It's called B+ Labs. And this is where the blood will be transferred and the plasma will be separated and prepared for shipment to Dublin and for shipment to another site in Milan, which is an high-level controlled biobank for being sure that nothing will happen to your samples. After that, there will be the biomarker analysis done at our labs in Dublin at Ulysses Neuroscience, and we will look at the proteins that I just mentioned to you. There will be integration with the data that we have already collected from other studies, and then we will show you the data. As I mentioned to you, Elpis is an hypothesis-driven. We analyze protein associated to a cellular system, which is called microtubules. Microtubules are very highly dynamic polymers. They're formed by proteins called tubulin, and the tubulin is formed by dimers, and one of those is called alpha-tubulin. The tubulin dimers, they polymerize and they form microtubules. They're highly dynamic structure, which means that they quickly switch from phase of elongation to phase of shortening. You can see, I don't know if I can play this little video. Yes, you can see it's just a video which is fictional, but it's giving you an idea of how dynamic the microtubules are and how fundamental this dynamic is to then finally create a neuron, as you can see there, that is healthy, functional, and functionally means that can develop, can maintain connection, can create connection, can remodel connection. Now, if the microtubule is malfunctioned, this cannot happen. And this is actually the case in CDKL5 deficiency disorder. And why? Because there is a direct link between the CDKL5 protein and the modulation of the microtubule dynamics. Microtubule dynamic is modulated by proteins that associate with the microtubule itself. They can associate on the surface of the microtubules, as you can see here. or they can associate on the what is called the plus end of the microtubules, which is the more dynamic one. CDKL5 is a kinase, so phosphorylate proteins, and phosphorylate these microtubule-associated proteins. By phosphorylating them, allows the MAPs, the microtubule-associated proteins, to modulate the system. CDKL5 also interact with CLIP170, which is another very important microtubule-associated protein. It's not a phosphorylation here. It's more of a mechanical interaction. So if there is a loss of CDKL5, there is a dysregulated microtubule dynamics. We have analyzed microtubule dynamics in my lab for the last 20 years, so we have a lot of experience. We have developed protocols by analyzing one protein, which is called acetylated alpha-tubulin, which is an alpha-tubulin modified with an acetylation in a position called Lys30. We have developed protocols where we analyze this protein in the brain of different species. When this protein is overexpressed, it means that the microtubules are less dynamic, so the neurons can function less. The recent discovery of a few years ago that we did in my lab is that this protein can also be detected in the plasma. And we have observed that the changes detected in the plasma did correspond to the changes we can see in the brain in rodents. Indeed, our Elpis did start. It's a wonderful example of a translational study. It started with a preclinical observation in CDKL5 knockout mice. CDKL5 knockout mice and the females, which are heterozygous, are mice, the males that don't have completed a protein because the gene is not expressed, and the females have only one copy of the gene that is functional. When we looked at analyzed the acetylated alpha-tubulin in both the males and the females in the brain, we saw an overexpression of this protein, indicating therefore decreased microtubule dynamics, altered neuronal function. We also found this overexpression mimicked, reflected in the plasma, as you can clearly see here, both in the male and the female. What is very important is that when we reinserted the gene using a gene therapy in the mice, in the knockout mice, we have seen that this overexpression of acetylated alpha-tubulin was rescued, which means that there is a direct pathogenetical connection between the overexpression of acetylated alpha-tubulin and the disease itself. So this was a very important data that pushed us immediately, together with the Lulu Foundation, to start Elpis and to start analysis in clinical samples. What we have found so far, and I'm very happy to share this data with you because are the most updated one, is with 49 CDD patients. You can see that we have confirmed what we have detected in the rodents. So we have a very high and consistent overexpression of acetylated alpha-tubulin in the plasma of these patients. It is also important that this biomarker doesn't change with the age. It means that it's stable. And this is what you want from a biomarker. We have also looked at other biomarkers, as I told you, to have a more complete picture of what is going on. Neurofilament light associated with epileptic seizures is increased in the plasma of CDD patients. We have also looked at BDNF. In this biomarker, we didn't find any change in our patients. What is important is also the immune system, as I mentioned to you. And we are getting another extremely consistent, as much consistent as acetylated alpha-tubulin, is an inflammatory marker. It's called IL-10, which is an anti-inflammatory, which is highly expressed in the plasma of our CDD patients. Some more recent reasons, so those data are not complete. We're also looking at other inflammatory markers, in this case pro-inflammatory, which means they induce inflammation. And we found increase in the plasma of the CDD patients of interferon gamma, TNF-alpha, IL-1 beta. Important to notice that those inflammatory markers are also acting on the brain. So they can also play a role in the disease itself in the brain. But the take-home message here is that the immune system in our CDD patients it appears to be altered or compromised in some way. That's all from my side. So thank you for following this webinar. I'm looking forward to meet all of you in June at the family event in Philadelphia. Thank you for your contribution. This is fundamental. Thanks to all of you, to all the patient community all around the world. Thank you also to the Rural Foundation for the generous support of this study. I will see you soon.
Thanks, Massi.
I'm going to make one quick comment. We have someone put something in the Q&A, said, we are ready to give samples. They're from South Africa, and said, but if necessary, they're ready to travel. So as always, our families are willing to step up and take part and do what we can to move things forward. Okay, take it away, Wendy.
Thank you so
Thanks,
much.
Karen. That's such a wonderful comment. And the family that put that Q&A in the chat, if you can get my accent, I'm also from South Africa. I'm from Pietermaritzburg. So nice to meet you. I'm going to share my screen because I've got a few slides. Can you see those? Mm-hmm.
Yes.
Okay, and I'm just going to put it into, see if I can put it into present. There, can we see that?
Yes, it is in presenter mode.
Awesome, great. So I'll just be very brief. Like what Massi and Ulysses are doing, we're also looking for biomarkers. We are a lab. We're based at in Australia, and I'm a professor at the University of Sydney. And our group is really focused on trying to find biomarkers for rare neurodevelopmental disorders. We've just recently finished a study on Rett syndrome, and we're about to publish the data. And now we're collecting samples from families and individuals with CDD in Australia. And thanks to Karen and Massi that we're collaborating with them so that we can increase our numbers so that we have a larger cohort. And the reason being is in Australia, we have a very small population. There are only about 25 million people in Australia, and there are not many families with CDD. We know there are about 44. However, I think realistically, as Massi showed in the slide, that we'll probably only realistically get about 10, maybe 20, maximum probably 30 samples. And so this is a really wonderful opportunity to be able to collaborate with the IFCR and Ulysses so that we can share our samples. Here is a map of Australia. My lab is based at Kids Research, which is at the Children's Hospital at Westmead, which is in Sydney. And you can see the front picture of the lab there. The reason why we need to look for biomarkers is in order to increase the number of treatments that are available for children with rare neurodevelopmental disorders like CDD. And biomarkers are like a blood test that tells us what's happening inside the brain. As Massi said, we can't analyze anything that's in the brain, but the blood can actually help us identify what's going on in the brain. So it's almost like a surrogate. And what biomarkers can do is help us track disease. So how your child's disease is progressing. It allows us to understand the biology as well, and also so that we can test treatments. You probably you may be wondering what on earth a biomarker is and why it's so important. And the bottom line is it's like a biological fingerprint. It allows us to understand exactly what's happening in your body and your child's body at one time. And the focus of our research is to look at biomarkers in a holistic way. So instead of being targeted, like Ulysses is doing, which is absolutely a focused way in which to identify biomarkers. What we're doing is we're taking a more holistic approach, looking at the blood and looking at proteins, metabolites, and genes and fats or lipids inside the blood to see if we can find new biomarkers things that we've never thought of before. And this can help us understand the disease as well, and then also lead to new treatments. So between what Ulysses are doing and what we are doing, it's a synergistic approach to have a targeted approach, but also a more holistic approach to look for novel biomarkers as well. And why are they most important? Why are they so important? And the reason why they're important is that there are a lot of new therapies that are being developed. And we as scientists, researchers, we're really good at understanding disease and coming up with therapies, but we need to be better at how to analyze how effective the therapies are. And that's what biomarkers can do. So, for example, with clinical trials, a good biomarker would really, really fast track clinical trials because it makes it more accurate. It allows us to say, we're testing a new therapy. How good is it? And in order to do this, we can use biomarkers that we can find and look in the blood to see how well the therapy is working. And of course, the families are so important, and you play such a crucial role in this research. And it's the only way that science can move forward is because families choose to partner with scientists and with companies like Ulysses. So in order for us to be able to identify a true biomarker, we need a lot of samples. Every sample helps. The more samples we have, the bigger cohorts that we have, the more specific our biomarkers will be. And just to give you an overview of what we're doing in the lab, very, very similar to what Massi said, is to look at to gather together individuals with CDD and to have neurotypical controls. Neurotypical is the same as healthy in Australia and in the US as well. As I said, we only have 44 families in Australia, and it'd be great to be able to include samples from the US and from around the world as well to increase our numbers. And then after signed consent, we'll take blood, they'll be sampled and analyzed. I've got written here omic analysis. What we're doing is an omic analysis is any word that ends with omic. So for example, we're going to be looking at transcriptomics, which is the genes, proteomics, which is a protein, metabolomics, which is the metabolites, lipidomics, which is the lipids, and through a clever bioinformatic pathway and with very, very clever bioinformaticians, what these bioinformaticians do is they put all this data together and look for biomarkers holistically, not just a gene or a protein, but look at them all together to identify biomarkers and also biomarker signatures as well. So it's a very AI and machine learning process. We're using AI to determine what biomarkers will be best and how we can then move forward with these to use them as disease drivers to understand the course of disease, to understand if existing treatments are working, and also for clinical trials to understand if a treatment that is being tested in a clinical trial is actually effective or not. And this is just my last slide, just to show the flow how blood samples can be used for biomarker discovery that can lead to improved clinical trials and then lead to better treatments. I've just left a slide on here at the end because these slides will be distributed. My contact details are here, my email address, my University of Sydney profile, my Children's Hospital at Westmead profile, and just a quote to say that many breakthroughs in rare disease research have only been possible because families choose to participate in studies like this. So thank you very much. And I'll be in Philadelphia in June, so it'll be great to meet you all. And lastly, this is just a slide of some of my team, and happy to answer any questions if there are. I'm going to stop sharing now.
Thank you so much, Wendy. Thank you both to Massi and to you, Wendy. I appreciate you taking the time to explain this to the community. We do have another question. It was at the start of your talk, Wendy, and I think you did answer. It says, are the biomarker studies intended to find drug treatments? And I think you did a beautiful job of explaining that it can be a mechanism to use to measure treatment. It can help us identify potential targets if you want to speak to anything else, but I do think what you presented answered that question.
Yeah, Karen, if I can add on that question,
Sure.
especially for the Elpis study, which is hypothesis driven, and is hypothesis driven for a reason. And sorry if you don't see my video, but if I switch on my video, I will lose the I will lose the connection.
No problem.
So the microtubule and the microtubule proteins that we are analyzing are also a target, and it's a target that is druggable. And in parallel, we are running another study, which is called SEMA-L, which has been presented for the first time at the CDKL5 Forum last year. So we are developing small molecules that do target these alterations that we've seen in the plasma of CDD patients, and we have actually finished. We have now finished our first round of experiments in the mutant CDKL5 mice. The results are extremely promising, and I hope to show them at the next forum in London this year. So yes, biomarker study can help developing new drugs.
Thank you. Let's give a couple more minutes. If anyone else has any questions, please put them in the Q&A. Again, I just want to thank everyone for joining. I want to thank the participants for their presentations. I am so excited about Family Conference, and to me, there's nothing better. It's my favorite thing. We only have it every 2 years. I wish we could do it every year, but it's a lot of work. So it is every 2 years, but I look forward to seeing everyone there. Does anyone else on the team have anything they want to say?
Just thank you for joining us. Really appreciate it.
Okay. I don't see any more questions in the chat, so I think we'll be respectful. We'll wrap it up in an hour, and we will see you all in Philly. Thank you, everyone.
Thank you.
Thank you.
Thanks. Bye.
Go. Have a great day.